Okay, I'm glad that I have the opportunity to present you some work I did together with my PhD student, Michael Welter, or better, he did together with me. And I thank the organizers, in particular Carsten, for giving me that opportunity. Actually, we switched scales now from colloids to actually to tissues, and in particular to large tissues, which can only survive when they are provided by nutrition, which happens in living organisms by blood vessel networks. And here I focus on this blood vessel network and the vascularization patterns of and fluid flow in growing tumors. So essentially, what I'm interested in is how a growing tumor transforms a well-organized hierarchical arteriovenous network into something messy like this. So this is a typical tumor vasculature looked from outside. Here inside you have the tumor where the star is, and you see that the hierarchical organization is totally lost. The vessel density is highly increased. So here, essentially in this network, you have a more or less homogeneous coverage of the tissue by capillaries, the lowest levels vessels, which essentially emit the nutrients and the oxygen. I mean, otherwise it wouldn't be. First of all, it would be uneconomic to put more vessels than absolutely necessary into a tissue, and it would be lethal if there are too few. So essentially, when you look into the healthy tissue, you will see that there are capillaries, all depending on the tissue, 50 to 100 micrometer. In particular, densities, of course, in brain, where the capillary distance is 50 micrometer. Yeah. And here you see that the density is actually even increased, but this is not always, not only there the case, but also inside. So this goes in the literature under the name angiogenesis, and maybe you know that angiogenesis is sort of the second basic pillar of, or better, the prevention of angiogenesis, the second pillar of any cancer treatment. The first pillar is attacking tumor cells directly. So for clinicians, this is very interesting, that also when you, as soon as a physicist wanted to study tissue growth beyond a particular size, let's say a millimeter or so, you have to consider the fact that the blood vessel network has to keep up. So first away, the course of the talk is, first I will confront you with some experimental data, which you might not know. You might know maybe the cartoon you find everywhere. For instance, in the in the Cancer Bible by Weingart, where you have here a vessel and here a tumor, and angiogenesis is essentially something that proceeds from this mother vessel towards the tumor. This is neither true nor wrong. This is a cartoon, just in order to provide sort of the picture that you have two different things, namely a blood vessel network and a tumor which is stimulating the blood vessel network in order to grow towards it. In reality, it's much more complicated, as I will show you in the next slides. Here you have pictures, actually stained slices of brain tissue, red tissue, red brain tissue, where sort of the blood vessels are sort of the normal. This is the normal tissue, the normal cells, sort of stained like in gray. Tumor cells give this reddish color, and black is endothelial cells, which cover, which align the lumen of vessels. So you see that the tumor grows within a well-vascularized tissue. Of course, this has to be this glioma. They grow in brain, and brain is the best vascularized tissue in the body. So after a week, you see that things happen, namely the tumor has grown, as you can see, but simultaneously you see that the density of vessels in the perimeter of the tumor has increased significantly, and inside it has reduced a little bit, and the black dots became thicker. So it essentially the radius of the vessels increased as compared to the capillaries in healthy tissue. Now, after 4 weeks, of course, this is all different rats. Of course, after you have to obtain this sample, the rat is dead. But it's the same tumor. So after 4 weeks, the change is even more drastic. Here you have an incredibly large sort of vascular cortex, which is essentially what we saw in the picture before, this mass. And inside, you see only a very few very thick vessels which are surrounded by viable tumor cells, so like a cord, and sort of within a sea of necrotic tissue. So it's that. Simply oxygen that is emitted secreted from a vessel like this here diffuses 150 micrometer, and because it gets consumed by tumor cells and then it's off. So there's death here beyond this typical length scale. So when you look at this, it looks as if the growing tumor has pushed the vessels outside. But this is not what happened. Actually, the tumor cells just grow around it. So what actually happens is that there's proliferation, so I will show you, of vessels outside, and degradation or regression of vessels inside. That is demonstrated in this series of pictures, where you see here a thick vessel surrounded by endothelial cells and also by pericytes in an early stage, but progressively inside the tumor, which gets progressively degraded and then vanishes. So there is essentially what you observe is, and you can also measure it more quantitatively, is vessel proliferation outside and degradation or vessel collapse inside. So that the emerging picture is essentially this. This here is again, these are the data by which experimentalists attain the data. The message I have tried to convey that sort of in a distance larger than 100, 150 micrometer away from a vessel, there is no oxygen anymore, and the tumor cells hardly survive. Of course, there are tumor cells they can switch their metabolism to glycolytic pathway, but anyway, the glycose has also to come from somewhere. So, and then there's also connected with that is also this the pH issue, which I do not want to mention here. And beyond that range, the tumor cells, they suffer, or any other cells suffer hypoxia, so the lack of oxygen, and that triggers a change in the gene expression of tumor cells. Typically, it's called the switch of the HIF, hypoxia-induced factor. Factor means you do not know exactly whether it's one protein, several proteins, or gene expression pattern which changes. So which then leads, among other things, to the production of growth factors. For instance, a vascular endothelial growth factor, which triggers endothelial cells to migrate and proliferate and so on. So the emerging picture is essentially this. This is sort of the healthy tissue with a typical vascular density, and inside this is tumor and also vascular endothelial growth factor. And after a while, it transforms to something where you have here this vascular cortex with increased vessel density, these thick threading vessels with viable tumor cord and a necrotic core. Okay, the formation of new vessels induced by growth factors, here in this case by BFGF, can be demonstrated in vitro. Here you have essentially fibrin gel plus this growth factor, and beads, microbeads, coated with endothelial cell. And since there is this growth factor, these endothelial cells, they're stretched into the gel and migrate through the gel, and from time to time they meet, like this here. Yeah, these two have met, and they perform something which is called anastomosis, so they unite. When I think, let me see, there is one here. This guy did not find something, yeah, here and here, so he retreats again. So it's really something. In vivo, the tip of the sprout doesn't need to hit the other vessel directly. In particular, in 3 dimension, that becomes difficult. But there are these. The tip has these philopodia extending into the surrounding extracellular matrix, which have sort of a radius, so cover a radius of 20 micrometer. And when you consider the fact that the typical vessel-to-vessel distance or capillary-to-capillary distance is 50, 80 microns, I mean, the probability that you hit the next vessel is very high. So you do not know. You also do not need to consider chemotaxis. It's just very short migration. You hit it, and that's it. Okay. So there are a couple of other facts, but at some point I have to come to our model, and this is essentially trying to incorporate these observations. It contains essentially the 3 basic modules. Of course, you need the representation of the vessel network, which is dynamically changing under different conditions. You need a tumor model. Yeah. It's essentially a growth process, nonlinear, with proliferation and death rates, which depend on oxygen concentration and pressure and things like this. And then there has to be communication between these 2 units, between the tumor and the vessel network. Essentially, the tumor talks to the vessel network via the concentration field for the growth factor. When the growth factor is high, tumor cells proliferate. When the growth factor is high, vessels initiate sprouts, yeah, which then try to migrate through the extracellular matrix and try to find a connection partner in order to induce flow. And when it's slow, nothing happens. And growth factor is also induced via oxygen. Oxygen is produced by the vessel network, so all of these vessels sort of they carry a blood flow, which you also calculate. So we assume it to be pipe flow, ideal pipe flow. And whenever you have a perfused vessel, it serves as a source for the for the oxygen. And when oxygen is there, this is just one. It could also be some other nutrient, but this is the most prominent one. When tumor cells feel that there is enough oxygen, they proliferate. When there is not enough oxygen, they stop proliferation, and at some point they will die, which then gives rise to contribution to the necrotic tissue. So essentially, here you can take whatever model you like, including or excluding morphological instabilities. Currently we take a mixed phase model describing a mixture of incompressible liquids with high viscosity, normal cells, necrotic tissue, and tumor cells, and proliferation and death rates, which depend on the presence of oxygen, the concentration of oxygen field, and so on, and the pressure, so density, and things like this. I have transparencies if you ask, but this is then not what I want to tell you. I would like to focus on this one here because it's about the blood vessel network. So essentially, when you have this network, I just remind you again, we assume ideal pipe flow within each link of this network, where the flow rate is proportional to the pressure difference between 2 junctions, and also proportional to the force power of the radius and inverse proportional to the viscosity, and there's a shear force. And you have mass conservation at each node. So essentially, the vessel network is something like a random resistor network. It's only not electrical current, it's blood current. And this is here it's essentially Ohm's law, where the resistance is given by a number which depends on essentially on the fourth power of the radius. One should not forget this analogy when one tries to understand in particular how, for instance, increasing the permeability of the vessel wall, what kind of consequences that had on the flow of fluid outside of the vessels in the interstitium. This is called the interstitial fluid. This can very easily be understood by just remembering when you increase in a series or a network of resistors, when you increase this resistance or that resistor or decrease it, what then happens actually. There are some false conclusions in the medical literature about this. Okay, and then you have equations for oxygen and growth factors and so on, but I do not want to mention that. I just want to repeat again the sort of the microscopic processes, which are essentially stochastic, concerning the vessel network. So you have this, when you have an existing perfused vessel network, you have sprout initiation when the growth factor field, close to the tumor, which is in yellow here, is larger than a certain threshold. Then the sprout migrates in some direction. Actually, we initiate the direction into the direction of the gradient, and afterwards we do not change because the density is usually that high that there's not much sense to change, to incorporate this chemotactics. And this happens with a particular rate. Then there is vessel wall degradation. You remember that inside the tumor, vessels degrade, become more unstable towards collapse and regression. Collapse sort of depends on whether the vessel wall is sufficiently degraded, unstable, and the shear force of the flow inside the vessel is below a particular threshold. So when, for instance, when this is the case here, this one collapses. And there is one also a very important issue you have seen in the pictures before, that the vessels became thicker. This is not by vasodilation, because this is significantly thicker. There is a process which is known as essentially inside the tumor there is still growth factor present, but the angiogenesis is sort of switched from angiogenic sprouting to circumferential growth. So you have still proliferation of endothelial cell, but they do not go into the tissue anymore, but they just enlarge the perimeter of the vessels. That is an experimental observation, and also the proteins involved in this process are identified. Okay, and then you need an initial network. Of course, when we started this thing 6 years ago, I started with a square lattice, of course. And then, of course, when you try to induce a blood flow into a square lattice, then you get all kinds of artifacts, namely a global flow, for instance, from the upper right corner to the lower left corner. So, in order to avoid all these artifacts, and in order to say something physiologically relevant, you have to have an hierarchically organized arteriovenous network, and since one cannot buy one, essentially there are data on the market or are published on arteriovenous networks in real tissue, but usually people sit very much on these data, so we have just to generate our own. Essentially, you have two trees. The problem is you have one. It's very easy to generate one tree, very nicely with all kinds of features, and fractal dimension is correct and so on. But now you take a second tree. This is the arterious tree, and this is the venous tree. Now you have to interdigitate these two in such a way that the whole volume is more or less homogeneously covered by connections of the lowest level, because that is what nature actually achieves. The homogeneous supply of oxygen and other nutrients to a piece of tissue. So this is a non-trivial model, but we have one which does that. It's sort of slightly motivated by what actually happens in nature, but it's not completely physiologically. In the end, we have a hierarchically organized arteriovenous network which fulfills this criterion. So essentially, in 2 dimensions, it looks roughly like this. We have here 2 arterial branches and 2 venous branches, and put sort of the in a different realization or different run, with 1 arterial branch here and 2 here, gives a slightly different network. So this is a stochastic process, and you have to average over different realization of that. Put a tumor into the middle, a nucleus of let's say 100, 200 micrometer size in yellow. The green is the growth factor concentration high. Then what happens is that this thing grows, and you see already that the incorporated elementary processes give you exactly sort of what I tried to emphasize in the first experimental transparencies: increased vascular density at the boundary, decreased vascular density inside with necrotic regions, and dilated vessels which are surrounded by a cuff of viable tumor cells. While in a 3D simulation it looks roughly like this. Where we have sort of cut open the tumor here, just that you can look inside. You see that essentially what looks like pushing the vascular cortex away from the tumor is simply what actually, here you have the proliferation of enhancement of vascular density induced by the growth factors, and inside you have a collapse of the vessels due to the vessel degradation and low shear forces. You can ask why due to these processes the center is not empty. This is more or less due to the fact that the amount of blood which is pumped into the tumor is more or less a prescribed value, which is constant from outside. And when all these vessels here, when they collapse and reduce the vessel density, then of course the shear force in the remaining vessels is very, very high. And that is good for endothelial cells. They like to feel this because they have the glucocalyx hanging into the bloodstream, and then they don't feel like collapsing. So this is expressed in very simple words. Here are a few snapshots where you again see what actually, and when you show you these things to people working in experimentalists, sort of medical people working in this field, they feel that this is pretty realistic visualization of what is actually going on in the tumor. So when you can do this, you can then it's just when you have that, you analyze that quantitatively. For instance, by measuring vessel radius, vessel density, blood flow, shear force, growth factor density, and oxygen concentration as a function of the distance of the tumor center. And here, in particular, in vessel density, that here you see that essentially this is sort of is a function of the distance of the tumor center in millimeter, how at different times, 100 hours, 200 hours, and so on. Oops. And you see that oh, my battery is low. Wow. So you see that here, this is sort of this bump is the vascular plexus at the periphery, which pushes more or less linearly outside. So it's in the linear growth regime of the tumor. The vessel radius increases inside the tumor. The tumor density is always given here by the broken lines. It's, of course, is low in the middle because there's necrotic region. So I just this drop here. Yeah. But nevertheless,
there must be a very broad distribution of vessel radii, actually. So when you plot vessel radius, you mean vessel radius?
Actually, not so. I mean, essentially, they grow until a particular maximum radius. Maybe you see, essentially, here they have all the same. In the boundary, fluctuations are high.
And
yeah.
In the healthy tissue, the fluctuations are enormous.
Of course, yeah.
Oh, but you say, so one characteristic of the cancer cell is that of the tumor region is that the vessels are all about the same size.
Yeah.
And that comes out.
I mean, in this region where you have necrotic.
Right,
I mean, here
right, in the necrotic region.
Yeah. Which corresponds also to what I've shown in the experimental data before. So, in spite of the fact that the vessel T drops significantly in the tumor center, the blood flow goes up like hell in the tumor center. How comes that? Simply because blood flow goes with the force power of the radius, yeah, and that makes it essentially this incredibly high average vessel radius in the tumor makes up for the loss in density. Shear force is also low inside, but does not drop to zero. So essentially, as soon as you have here many vessels at the periphery, shear force drops simply because the same constant amount of blood this blood flow has to be distributed over many vessels. So the shear force drops. The vessels are likely to collapse, and the same is the amount of oxygen essentially also drops here. So essentially these data represent this typical compartmentalization of tumor vasculature, which is observed in many different solid tumors, and it gives already an answer to a mystery that, for instance, people working with breast cancers have observed, namely that they by some physical chemical methods, they can measure the relative blood volume in tumors and also the oxygen content in cancer tissue, and they observe that the relative blood volume is increased a lot by a factor of 3 in tumors, which is an indicator of a malignant tumor due to vascularization, but somehow the oxygen content is low. So how comes? They cannot explain, but with this feature essentially you can easily explain, because essentially the relative blood volume goes with the square of the radius, and the secretion of oxygen through the vessel walls is proportional to the radius only. So when that means when you have a situation like this, and the radius goes up like this, then essentially and the vessel density goes down like this, then essentially we have the situation that oxygen is low, but relative blood volume compared to healthy tissue is high.
So that means the blood that's carried into the tumor has the same oxygen content as the blood that's carried in anywhere else in the body.
Yes.
But when it leaves the tumor, it's still got much of its oxygen. It doesn't actually leave much behind.
So sort of the secretion of oxygen or any diffusible substance is proportional to the surface of the vessel. Yeah, and that is
So I'm just saying that if less oxygen gets into this tumor, that means the blood that comes out has to be oxygen rich relative to the blood that flows into the healthy tissue.
Essentially, yes. Yeah. But that's true. But on the other side, it could be that sort of the stretches in which oxygen is secreted are a little bit longer, because it's within the tumor. It's hard to say what is artery, what is a vein, what is a capillary. So essentially, and all the rest. Yeah. Okay. Just as a side remark, I really didn't want to talk about percolation or self-organization or something buzzwords like this, but I just want to say that actually this one here, Rakesh Jain, he's a sort of the guru in tumor angiogenesis and analysis of tumor blood vessel network, and they published it some time ago, sorry, no, nearly 20 years, something where they analyzed photographies of carcinoma vasculature, and via box counting they found a particular fractal dimension, and they found this sexy and published in PIL together with an explanation for a particular number that comes out. I call it not fractal dimension, it's a number which comes out of the box counting procedure 1.89, and provided sort of an explanation that this could be invasion percolation, because these sprouts, they go into a disordered environment, and that is there are some spots in the locations in the tissue which are easier to penetrate with for sprouts than others, and so that gives them rise to first 8 point. This is not invasion percolation. This number is normal percolation, but this is a minor detail. I think the physical explanation is simply different. In particular, I guess you understand that also now on the background of the figures which I've shown before. The vessel growth is confined to the perimeter, and the morphology, and whenever you want to characterize that by a number, is determined by the vessel collapse within the tumor, which is then similar to conventional percolation, where you remove with particular probabilities links in a network. And but conventional percolation is only critical at a particular point. But due to this mechanism which sort of stabilizes the flow correlated stabilization mechanism inside the tumor, because the shear force is there very high and collapses probabilities is reduced, that you do not have to trigger a particular number in order to get, like say, complex or fractal looking structures, but it's organized by itself. And also, you have to consider when you do this box counting procedure, the smallest boxes you have they have the size of the vessels, and then you increase these boxes. So essentially, you measure the fractal dimension of the tumor and not of the vessel. Yeah, wow. It's a one-to-one relation where the tumor is there, tumor is where vessels are, and vice versa. Maybe the rest is necrotic. Okay, just this is a side remark. The formation of hotspots is also an issue in the medical literature. This is just a phenomenon that you have accumulation of blood at particular points in the tumor, which you can also observe in the dorsal skin fold chamber, with which experimental surgeons they do essentially analyze it via an intravital microscopy. You also see these inhomogeneities, and we find essentially that the hotspots are located. This is increased. I call it increased spots or locations or clusters of increased vessel density. They are correlated with the pressure gradients of the initial network. When you just coarse-grain this picture, you calculate the pressure differences, and then this region here has high pressure gradients, and this here lower pressure gradients. And that makes sense, because collapse of vessels is related to shear force. Shear force is related to the pressure gradients. So whenever you have a shunt between, for instance, an arteriole and a venule, there's high shear force in the emerging vessels, and this vessel is less likely to collapse, and this is sort of the mechanism which leads to accumulation of vessels. Drug flow. So you can we can inject into the blood flow of an artery a bolus for some time, and it has once speculated that because the tumor vasculature is bad or something the drug distribution is also bad. But this is not true, as you see here when you inject the drug, which is just in red. After a second, this is seconds here. After 5 seconds, there are only some individual nests remaining. In everything else carries sufficiently high drug concentration. This is also easily explainable because, of course, we have here only those vessels which survive, which are well perfused and have high blood flow. Okay, there are only these nests where you sort of there's like dead ends where drug concentration is low. But therapies often fail because the drug delivery into the tumor tissue or towards the tumor cells is bad. And then there came up the speculation that this is linked to elevated intravascular interstitial fluid pressure. So essentially, this is the situation. Here's a blood vessel, and it emits a drug, and of course the concentration falls down. This is a picture of doxorubicin, which is a pretty common anti-cancer drug, and you see this the blue, the red is vessels, blue is the concentration of doxorubicin, and here are the hypoxic regions, and so here is sort of something which has not been reached. And it has been speculated, again by Rakesh Jain, that because the tumor doesn't have lymphatics, and the tumor vessels are very leaky because they grow so fast, so they have lots of holes and so on. The fluid pressure in the extracellular matrix, so in the interstitium, so the space between these cells, his fluid pressure is high, and that reduces convection, and that inhibits the distribution of macromolecules. They even suggested first, as a anti-cancer therapy, first to normalize the tumor vasculature so that drug delivery is improved. Well, as somebody who took the analogy with the resistor network serious, he knows already there's something fishy in this argument. But we incorporated interstitial flow into the model, essentially via Darcy's law. So each leaky tumor vessel is a source of interstitial fluid, and that is then essentially driven by pressure difference and proportional to permeability of the tumor tissue. And you have to solve this in the whole region within the tumor. So first you have to grow your tumor vessel networks. You have the tumor with necrotic regions and so on and so on. Then you solve for a snapshot the equations for the convection diffusion of interstitial fluid, and that is what actually comes out. You see interstitial fluid pressure. It's high here, much higher than here, as expected, of course. Fluid source strength is where the thick vessels are. And this is here the X component, the modulus, and the outward component of the flow field of the interstitial fluid. And you see immediately that the flow field is not diminished inside the tumor. Here the flow field is low, but in general you have here an outward component, and this is high. And of course, the flow patterns are complex because leaky vessels cannot only emit fluid, but of course, when the pressure is low, it can also subtract. So it's really, again, a complex resistor network with certain permeabilities. If you analyze this quantitatively, This is sort of the distance from the tumor center. Negative means inside the tumor. Positive means outside the tumor. The interstitial fluid pressure goes to the plateau, which is which is expected. But the interstitial fluid flow is by no means small here. Yeah. And with these variations here, we vary various permeabilities, vessel wall permeability, interstitium permeability, lymphatic permeability, because lymphatics are outside the tumor, and of course they have also you have to pass a particular resistance in order to, the liquid has to pass a particular resistance in order to go into the lymphatics, so they have also permeability. And so on. So then you find that interstitial fluid flow increases when any of the permeabilities increase. And just skip that. Maybe just, and then you insert sort of, within this flow field, you can of course then analyze how the drug, an inserted drug bolus, distributes, and it essentially also shows the same result, that it correlates positively with the permeabilities. And then the variations, essentially, when you increase here or decrease the permeability compared to the base case vessel wall permeability, then sort of the maximum, so the integrated amount, time integrated amount of drug is, when you increase the permeability, is higher, and when you decrease the permeability is lower. So essentially, decreasing vessel wall permeability decreases also drug delivery. That means the suggested normalization of tumor vasculature, according to again, does not work. And this was actually found out. So you have to imagine, he comes up with the idea that due to interstitial fluid, increased interstitial fluid pressure, drug delivery might be impeded. So try sort of angiogenic therapy first, with which you normalize the tumor therapy, and then that costs millions and millions of dollars. And then the only thing that you have to do, essentially, when you have a series of resistors, to think when you decrease one of the resistors, what happens? Does the current increase or decrease? So essentially, this was the mistake. Okay, let me just give you a conclusion of what I have done, and my time is up. Thank you very much.
So did I understand you right? You said earlier that you tend to see the biggest changes in the vasculature in regions where the original healthy network had high pressure
Gradients.
Pressure gradients, right?
Yeah.
So does that mean that there's that would seem to imply that there'd be a correlation between cancer and various kinds of vascular disorders? You know, you get blockages in the vessels or holes. You know, so people who are prone to strokes or blood clots or something would be more inclined to get tumors in those regions of the body.
This I don't know. These are local variations of the original vasculature. So imagine that you have a region in space where in the tissue you have here an artery which is close by to a vein. So now the tumor comes and grows over it, and sort of stimulates via angiogenesis a shunt between these two. So because the gradient is here between the sort of the blood pressure gradient is high. Yeah. And this shunt is most likely to survive because simply it has a high shear force. This concerns local variations within the healthy.
Right, but I'm just wondering if there are vascular conditions or diseases in which you're more prone to have such proximity of arteries and veins.
This I don't know.
So for the drug study you reported there, the decrease was assuming the drug just convected with the fluid rather than diffused. Is that what the results were for? I didn't quite understand. When you said you did the example of the drug distribution as a result of the change in permeability in the tumor,
yeah,
is that just assuming the drug was convected into that space?
it is a mixture of convection and diffusion. Of course, there is a sort of a parameter. It depends on the. There are various kinds of drugs. Some are heavier and some are lighter. So for the heavier drugs, the convection dominates, and for the lighter drug, of course, diffusion. And essentially, increasing the permeability, so means the amount of drug and of interstitial fluid, or fluid that enters the interstitium, has an effect on both diffusion and convective drug transport. And in both cases, when leaky vessels just increase the interstitial fluid flow. And
I think there's also another effect that the leaky vessels are more porous and emit particles of larger size than non-leaky vessels.
yeah, but that's true.
You can make a therapy that way,
Yeah.
which is actually in clinical trials for cancer treatment by doing precisely that.
That
Large
is-
particles that don't go in.
Yes,
They go in the tumor zone.
this is totally reasonable, but then somebody would say this is outrageous. How can you suggest something like this? Because
Oh,
it increases the chance of metastasis when you
No, they're in clinical trials. People have been lives have been saved by this therapy.
Ah, I see. but when you increase the permeability of the vessel wall, you simultaneously increase, of course, the probability with
But
which
these are tumors that already exist, that have already increased the permeability, they're leaky. So a bigger object can't get out of the rest of your blood vasculature, but can get into the tumor only.
Yeah.
So you load that big object with bad drugs, it doesn't affect you anywhere else, it just kills the tumor,
Yeah.
and it works.
If there are no further questions.